Welcome, everyone. We'll get started at 4 p.m. Eastern Standard Time. Thank you all for joining.
Hi there, everyone. Thank you for joining us today. This is our third installment in 2024 of LBDA's Community Webinar Series. My name is Keith Fargo, and I'm the Director of Scientific Initiatives here at the Lewy Body Dementia Association. It's really my pleasure to be here with you today to moderate this webinar. Today's topic is one that is of very high interest in the scientific community, as it no doubt is to you. As the webinar proceeds today, you may find yourself intrigued by this kind of research and wonder how you can get involved in research on biomarkers. If so, I would encourage you to visit our website to learn more about research studies that are happening today. And as a matter of fact, there is one on one of the biomarkers that you're going to hear about today. In particular, this study called SYND, and we're going to drop a link to that in the chat for you so you can find that. That SYND study is currently seeking participants with mild DLB to study one of the synuclein biomarkers in the skin, and you'll hear about that marker today. So Rose, did you get that in the chat? I'm going to assume she did. Thank you very much. I do see that there. So before we get started, a few words about LBDA. We are a national nonprofit organization whose mission is to optimize the quality of life for those affected by Lewy body dementia. And we do that by accelerating awareness, advancing research for earlier diagnosis and improved care, and by providing comprehensive education and compassionate support. A few housekeeping items before we jump in. First of all, you're welcome to submit your questions at any time. Please submit them through the Q&A button, though, rather than through the chat button. Following the presentation, any of the questions that came in through the Q&A will be gathered into a pool of questions, rather. And I will select some of those questions to pose to Dr. Poston. And we'll do that for about 15 minutes for the Q&A at the end. This presentation is being recorded, and an email will be sent to those who registered with the link to view the recording in about 1 week. To view any of the recordings that you may have missed from earlier webinars in our series, you can visit our YouTube channel, which is on LBDA TV. And I see Rose dropped a link into the chat for that as well. When you leave the webinar today, a brief survey will appear that's going to ask you for your feedback about today's presentation. Please do take a moment to complete that survey. That really does help inform our future content, so we would really appreciate it if you could take the time to fill out those brief questions. And speaking of questions, let's take a minute now to find out who's joining us in the audience today. Rose is going to launch a polling question, and you'll be able to answer that on your screen. Wow, lots of answers. Okay, we're closing on over 200 answers here. So thank you for everyone who's answering. It looks like most of the folks who are joining us today are either someone with Lewy body disease or someone who is a care partner for someone with Lewy body disease. Looks like that's making up about 65% of the audience. And then we have friends and family members of people with LBD. We do have about 10% of the audience actually is healthcare providers today. And then about a similar number of researchers, and then, say 5 or 6% other. So thank you very much for taking the time to complete that poll so we have a better sense for who's joining us today. Now I'd like to introduce today's speaker, Dr. Kathleen Poston. It's a real treat for us to have Dr. Poston with us today. I really only have time to just mention a few things about her impressive bio. Dr. Poston is professor of neurology and neurological sciences and neurosurgery at Stanford University Medical Center. There, she leads the Udall Center of Excellence for Parkinson's Disease Research, the research education component of the Stanford Alzheimer's Disease Research Center, and the Lewy Body Dementia Association Research Center of Excellence. Dr. Poston's research focuses on the development of biomarkers to advance our understanding of neurodegenerative disease. And her current work is focused on cognitive decline in people with Parkinson's disease and dementia with Lewy bodies, making her the perfect person to give today's presentation. So without further ado, I would like to turn it over to Dr. Kathleen Poston.
There we go. I shared my screen before I unmuted. I should know the right order to go in. All right. Thank you all so much for joining today. It really is my pleasure to have the opportunity to talk to all of you about some very exciting developments that are happening in the field. And thank you so much to Keith and to the rest of the team for inviting me. I'm just start off by saying that this is a very fast moving field, which is excellent because we need new information and we need new information quickly. But it is a lot to digest, and so I'm going to try to bring it down to some bite-sized information that hopefully you can all walk away with understanding a little better into how researchers are now seeing in some way Lewy bodies in the living brain. So I included my disclosures just because I do want to point out that I am on the scientific advisory board for one of the companies that I will be talking about today, Amprion. And so I'll just make that clear so everyone knows exactly where all my disclosures are. Okay, so let's start off. I'm going to start off by just giving you a snapshot of the pathology and the way that we think about neurodegenerative diseases. And then I'm going to talk a little bit about how we currently diagnose people, and I'm going to keep that very short. That could be a whole lecture in and of itself, but I just want to be able to contrast that with where these new biomarkers might come into play with our current approach to diagnosis. Okay, so first of all, what is Lewy body dementia? It is a neurodegenerative disease. That means that it is a disorder in which the neurons in the brain are affected and become dysfunctional and die over time. That's what a neurodegenerative disease is. There are a lot of neurodegenerative diseases, and this slide is meant to slightly overwhelm you with all the different kinds. But what I wanted to emphasize here is that we cluster the different neurodegenerative diseases right now according to the type of proteins that we see change in the brain at autopsy. So after somebody has a neurodegenerative disease and they die and they donate their brain to research, when we look at their brain, what kinds of abnormal proteins do we see? So I clustered into just 5 different groups here. The ones that are squared in a blue color are the ones that are affected by amyloid. That includes Alzheimer's disease, but includes some others as well. The ones that are in kind of more the teal blue color are different diseases that specifically are associated with just the tau protein abnormality. This includes things like progressive supranuclear palsy and cortical basal degeneration. The ones in purple are the ones that are associated with a protein called TDP43, that's frontotemporal dementia. And then there's another group in green that are associated with another protein, FUS. And so I just wanted to say that the ones that are in red in the middle of your screen are the different neurodegenerative diseases that are associated with the protein alpha-synuclein. So you're going to hear that protein several times during this presentation, but that's sort of how we categorize these different neurodegenerative diseases is based on the proteins. So Lewy body dementia is a neurodegenerative disease that causes cognitive impairment and dementia in which Lewy bodies are what is seen under the microscope in individuals who die from this disorder. And so the 2 disorders that are associated at pathology with Lewy bodies are 2 of these synucleinopathies, dementia with Lewy bodies, and Parkinson's disease. Multiple system atrophy, or MSA, is also a disorder where synuclein is abnormal, but it's not Lewy bodies. So it's not included as a Lewy body disease. Just dementia with Lewy bodies and Parkinson's disease are considered Lewy body diseases. All right, so that's kind of where we're honing in our focus here. So how do we know this? When people pass away and they either have their brain examined for diagnostic purposes or for research, the pathologists look at it. And this is this little cartoon of the brain and different areas of the brain. And there are certain parts of the brain that are uniquely affected in individuals with Lewy body disease. And this is one of them. There are other ones. This is one of them. This is a part of the brain here. It's called the substantia nigra, which is Latin for black substance. As you can see in this non-Lewy body person, it's this nice dark black ribbon. This part of the brain contains a pigment, neuromelanin, which has melanin in it, so that's why it's black. And here, this person with Lewy body disease, it's kind of a pale color. And when you look at this part of the brain under the microscope in a normal, a person who does not have Lewy body disease, you see all of these pink dots. Each one of these pink dots is a nice, healthy, functioning neuron that was very healthy before the person died. And here in the person with Lewy body disease, you see very few of those healthy pink neurons. The majority of the neurons are gone and have died. So looking under a really, really high-powered microscope to the surviving neurons, you would find this Lewy body. So this big pink blob should not be in the neuron. This is not in the neurons of the people who do not have Lewy body disease. So these were first described in 1912. These were some of the original drawings by Dr. Lewy that he made, where he saw these inclusions under the microscope, and they eventually were called Lewy bodies. Fast forward to 1998, scientists discovered that the protein in these Lewy bodies was alpha-synuclein. So that's how we get from Lewy body to alpha-synuclein. So this was just, barely 15 or 25 years ago. It was identified that the protein that predominantly made up these Lewy bodies was alpha-synuclein. Now, alpha-synuclein is not a bad protein. It's actually a normal protein that's in all of our brains, and it serves really important purposes. But for whatever reason, and we don't fully understand why, we see that it has clumped together and formed these Lewy bodies in people who have Lewy body diseases and Lewy body dementia. And I just also want to say that we actually don't know 100% whether these Lewy bodies are the thing that cause the neurons to die, or whether they're just a byproduct that does nothing. So it's just kind of neutral, but it's there. Or maybe it's actually something that's trying to protect the neuron, and then it fails, and then that's why the neuron dies. We don't fully understand the relationship between the Lewy bodies being formed and the neurons dying. So that's an important distinction. We don't know if it kills them or not. But we do find these Lewy bodies all over the brain, oftentimes in people who die of Lewy body disease. But the key thing is that right now, without any biomarkers, the only way to make the absolute definitive diagnosis of Lewy body dementia is at autopsy. I can't see these Lewy bodies under any type of MRI scan or PET scan or brain scan. They're way, way, way too small. There's nothing that binds to them in the brain that I can take pictures of, like on a PET scan. There's nothing that I can do right now with brain imaging that can look specifically at the Lewy bodies. All right, so what we do see, though, is, again, at autopsy, we can identify them. And this is a slide, a couple different slides of different people's brains at autopsy who donated their brains to research. And we actually can say how much of the brain has these Lewy bodies in it. And there's a pathological staging mechanism where they say, this stage 3 just has a little bit of Lewy bodies in it. Stage 4, it's all this brown stuff here. Through stage 5 and stage 6, we see the Lewy bodies everywhere. And so that's how the pathologist would say, this person has these different stages of Lewy body. If anyone's ever looked at an autopsy report of someone who's passed away, you'll see reference to this staging, which is called the Brock staging system. You can see the first name of the author here, Brock. And so this is the Brock staging system of Lewy bodies, which only can be used at autopsy. And it simply says how much of the brain do we see the Lewy bodies in with the staining. Okay, so that's all well and good. And then what they do is then they look under the microscope, and they see, the Lewy bodies, and then they stain them for the alpha-synuclein. And so basically you go from Lewy bodies to alpha-synuclein. Okay, so that's all well and good, but I'm not looking under the microscope in clinic, right? I'm diagnosing people every day with Lewy body disease in my during my clinic day, and clearly people who are alive have this diagnosis. So if that's the only definitive way of doing it, well, how do we do it in clinic today? And again, I'm just going to very, very quickly give you the highlights here. This could be an entirely other talk itself, so I'm missing a lot of the details, but just very, very high level, we have these clinical definitions for the diagnosis of dementia with Lewy bodies and a clinical definition of the diagnosis of Parkinson's disease dementia. And these rely on some key clinical features, such as, and just I'll give some examples here of visual hallucinations, fluctuations in cognition, this thing called REM sleep behavior disorder, having some spontaneous features of Parkinsonism, such as slowness, tremor, and rigidity, etc. And then there are some other biomarkers that can help with the diagnosis, but they're pretty they can be non-specific and they can be positive in other diseases. And right now, clinically, the only distinction we make between these two clinical definitions is that if the dementia occurs before or at the same time as the Parkinsonism, we call it dementia with Lewy bodies. And if the dementia occurs after somebody has already had a diagnosis of Parkinson's disease, we call it Parkinson's disease dementia. So it's just a timing issue, and there's a 1-year rule between these two, with a lot of continuity between them. But we use these clinical things, and we're pretty good. I mean, we're not 100%, but we're pretty good at making the diagnosis of dementia with Lewy bodies or Parkinson's dementia, but we're not 100%. And the other challenge is that a lot of people might have just one of these clinical features and not all of them. And it can sometimes take time for a person to have all these clinical features. For instance, someone with Parkinson's disease dementia, they might start off with just tremor, but not the slowness, stiffness, or walking abnormalities. And then over time, they develop these other things. So as neurodegenerative diseases change, we get better and better at the diagnosis, but early on, sometimes we can struggle with it. In addition, there are all these non-motor features, particularly within Parkinson's disease, that can overlap with other things, such as loss of smell or excessive sweating or blood pressure changes, which sometimes you can see in other disorders, which can also make the diagnosis very difficult. And then with dementia with Lewy bodies, again, the central theme is that people have to have changes in memory, but it can sometimes be difficult to differentiate this from things like Alzheimer's dementia, especially if someone doesn't have these more clinically defining features such as the visual hallucinations or the REM sleep behavior disorder. And so these are the types of things that can make the diagnosis a little more difficult in the clinical setting. And importantly, in research, you want to be 100% sure that every person you're enrolling in a clinical trial has the disease you're trying to target. Because if you have a lot of misdiagnoses in a clinical research, it adds a lot of noise. And anyone who's taken a statistics class knows that noise and variability is a terrible thing in statistics. And when you're trying to show that a drug is working better than a sugar pill, it's all about your statistical power. And adding noise into the clinical population that's getting enrolled really creates huge, huge problems for clinical trials. And so this has actually been one of the challenges in getting new drugs to patients over time, is that our clinical trials, we know a certain percentage of individuals are being enrolled who probably don't have that underlying pathology that we hope that they have, that Lewy body pathology, and we don't know that until the person passes away, which is many, many years after the clinical trial. So what is the status of assessing alpha-synuclein pathology in living people? Where are we in getting this pathologic diagnosis on the left side of your screen to a laboratory based? So just to give you a little bit of the context, traditional assays trying to detect alpha-synuclein in living people have been really, really bad. We've actually been able to measure alpha-synuclein in things like cerebral spinal fluid for a while in people, but it's a very, very messy, noisy assay. When I say messy in science, what I mean is noisy, has a lot of variability. It depends on, different aspects of who's doing the test and things like that. It's been really bad. It has not been something that we've even been able to use continuously in research. And I definitely, was never an assay that was even close to being usable in the clinical setting. So we really needed something new. So what I'm going to show you is 2 new ways that we believe have very good validity in identifying people who have Lewy body disease. And these are biomarkers. So I'm gonna really jump into a statistics slide here. I'm just prepping you right now, but I think everyone's going to understand what I mean when I walk you through it. So it's important to understand that a biomarker is trying to predict the reality. Okay. So I want you all to think back to the time when you took your first, COVID antigen test, right? Or if you got COVID when you took a COVID antigen test, and you put the little drops on the slide, and it either came up with 1 line, meaning it's negative because that's just the control, or 2 lines, meaning it was positive, right? So that antigen test was trying to predict who had COVID and who did not. And when we think about it, that was a biomarker. That was a diagnostic biomarker test. This is kind of what we're trying to do here, is have some sort of a test to predict who has the disease, in this case, Lewy body disease, and who's disease negative. So we use this confusion matrix to identify how correct we are. So if someone actually has the disease and their test is positive, we call that a true positive. If they have the disease and their test was negative, we call that a false negative. On the other hand, if they don't have the disease and the test is negative, that's a true negative. And if they don't have the disease and the test was positive, that's a false positive. And I'll just give one example. When I got COVID a couple of years ago, my husband had it for 3 days, and I had like a slight cough that morning. And so I was very, very high risk of being COVID positive. And so I took the test, and it was actually negative. But I didn't believe it because my husband had been sick for 3 days and coughing in my face. And so I was pretty sure I was going to be positive. And so at work, we could get the more specific, the better quality tests, which were the PCR tests. And so I got that like an hour after I did the antigen test, and that came back positive. So that means my first COVID test was a false negative. I actually had COVID, but the test came back negative. Now, the next day it came back positive. And so that's an example of a situation where early on in the disease, one of the tests had a false negative. So this is just really important to remember. When I think about biomarkers, what we're trying to do is predict reality. And sometimes it's very, very, very close to correct, but there are always these situations where it's slightly wrong. And one of the most important things we do in biomarker development is trying to minimize these false negatives and false positives, so that for most people, most of the time, the test is a true positive or a true negative. All right, but just it's important to keep that in mind when we talk about these, because I'm going to show you some examples of when these tests are not 100% right, and what does that mean in those situations. Okay, so biomarker number 1, the first one I'm going to talk about, requires a lumbar puncture. The name of this test is called the Synuclein Seed Amplification Assay, or SAA. It also goes by a couple of other names. I'll show that on the next slide. This particular biomarker right now does require a lumbar puncture or spinal tap. The physician removes a couple tablespoons of cerebral spinal fluid, and this is studied actually for not just synuclein, but can also be studied for other proteins, such as the proteins that I showed you on the first slide that are associated with Alzheimer's disease. So a lumbar puncture can test at the same time for Lewy bodies now and for Alzheimer's disease using a different type of test. I will say that they're trying to get this test to be valid in other types of fluids, such as a blood draw, but right now it is not. It's only available through a lumbar puncture. So how does it work? If you don't want to think about the science of it, then just ignore this part, but some of you might want to know, so I'm going to throw it out there. Also, this is known by 2 other very complicated names of RT-QuIC and PMCA. Whenever there's a new test, there's always a little bit of jockeying for like, what's the best name for it? Right now, the seed amplification test seems to be winning as the assay name, but I can't 100% say that that's what it's going to end up being. But nonetheless, that's what I'm going to call it the rest of the time, SAA or seed amplification assay. Okay, so we have this vial of cerebral spinal fluid, and our question is, does this cerebral spinal fluid contain the type of synuclein that sticks together and will form a Lewy body? That's the question. And so the way they figure it out is they put what's called recombinant synuclein, so these seeds of manufactured synuclein in there, and they put a fluorescence in there. And then they drop this fluid into all these different wells, and they go through this cyclic process of shaking the samples, fragmenting them, and then they elongate and form these long fibrils where if they're the sticky kind, they're going to create this fibril and stick together, and then all the fluorescence will kind of come together. And then they put that into a reader, and if there's enough of the stickiness that you form these long fibrils, you're going to get an amplification of the fluorescence. And that means, yes, the original CSF had these synuclein, had the sticky kind of the synuclein in there. Or if it comes out not detected, there's no fluorescence, that means the original synuclein did not. So an important point here, this is a yes, no answer, similar to your COVID antigen test. It either says, yes, you have COVID, or no, you do not have COVID. This simply says, yes, that synuclein was detected on the seed amplification test, or no, it was not detected. It doesn't say how bad it is. It doesn't say how severe. It doesn't say where it is. It doesn't say what kind of symptoms you're going to have. It's simply a yes, no test. They just see the amplification happen, and that means that it is a detected test. Okay, so how good is this? Well, what we want to get from is, again, this synuclein pathology under the microscope. And I included this little picture here because this is actually a picture of both Dr. Lewy and Dr. Alzheimer are both in this picture. We want to get from the pathology to something that we can see in humans. So let me show you some data. So this has been tested mostly in individuals with Parkinson's disease, but it has now been tested in individuals with dementia with Lewy body as well. This is some of the earlier Parkinson's data showing that in these people with a clinical diagnosis of Parkinson's disease, multiple system atrophy, or healthy older adults, that's what the HC stands for, that the people with a clinical diagnosis of Parkinson's disease, for the most part, all of them had the amplification, which was different than the type of amplification in individuals with multiple system atrophy, that other kind of synucleinopathy that's not Lewy body, versus the healthy older adults. And that this was one of the earlier publications in 2020, which showed very, very good separation between the groups. The bottom picture here is actually data from individuals who at autopsy were proven to have Lewy bodies. And so in the first bar here, you have what we call the non-synuclein pathology. So these were folks who did confirmed by the pathologist under the microscope, did not have synuclein in their brain. And one of them, you will see up here, actually did have a positive test. So this is a false positive test, was on one of them. But the majority who had synuclein in their brain, their cerebrospinal fluid that was taken before they died, so prior to death, all had elevated or fluorescence detected synuclein, except for one. There's a tiny little dot down there. You can see right at the line there. And so that is one false negative. So very, very high specificity. This last line here is amygdala only, which is sort of a unique kind of synuclein, which some people with Alzheimer's disease can have, which is a little bit different than the kind that is seen in individuals with dementia with Lewy bodies. But in the individuals with very clear cortical Lewy bodies, it was almost always amplified, and those without Lewy bodies, it was almost always negative. So this is a very, very high quality test. In addition, the research cohort I work with the Michael J. Fox Foundation, we have 19 individuals who have now passed away and donated their brains for research. And 19 have, at the time actually of their initial diagnosis, had a cerebral spinal fluid donated for research. And all 19 we have had confirmation on the assay. 17 of the participants who were SAA positive in CSF at the time of their Parkinson's disease diagnosis, all of them had confirmed alpha-synuclein Lewy body pathology at autopsy, and 2 individuals who were SAA negative at the time of the CSF were synuclein negative or did not have Lewy bodies. So again, perfect accuracy here, but this is a very small cohort. I am always very skeptical of anything that only has double digits in its research. And so this is a small cohort, and we need much larger numbers, because what if it's wrong like 1 out of every 200 times, right? You're not going to catch that when you only have 19. So you could just randomly have gotten the 19 times that it's perfect, but you need a lot more data to catch things that are inaccurate, you know, on the order of 1 out of 100 or 1 out of 200 or 1 out of 500. But this is very promising. Okay, moving on to biomarker number 2. So biomarker number 2 is peripheral synuclein detection. And this is a different type of assay called immunofluorescence. So this was first described in the literature actually, 10 years ago, where this group of researchers identified that taking a skin biopsies from individuals who had Parkinson's disease, they could see things that made them think they could identify synuclein in the skin. And this is an example. So in panel A, the green that you see is this is a skin biopsy. Panel A on the left side shows you what a stain, it's called PGP, which is a stain that that identifies a nerve cell. So all of these things you see on the left, all the green stuff, are different individuals who have, this is what their nerve cell looks like under the microscope. And on the right-hand side, the red, they are staining for synuclein. And you'll see the panels D and H, the 2 that say PD, alpha-synuclein. You'll see that the red overlays where the green is, whereas panels B and F, the red is not as bright, and it does not, you don't see it nearly as well. And so this didn't stain very well for synuclein, whereas in the PD patients it did. So this was their early attempts to do this. They have since really refined the approach tremendously. As I mentioned, this is called an immunofluorescence assay. This is what it looks like to get a skin biopsy. It's extremely easy to do. It can be done right in clinic. You don't even need a suture for it. And this is what it looks like when you overlay the red on the green. You see that you get these elongated synuclein in the nerve cells. Now this is a little bit more of a complicated assay in that it's a bit of a judgment call sometimes as far as exactly, you know, how much of the red stuff is really in the nerve. As you can see, you get a little bit of this background staining, but it's getting better and better. And to show you some data from this one, this is currently under investigation as well. They had a cohort of a little over 200 individuals with a clinical diagnosis of Parkinson's disease, multiple system atrophy, dementia with Lewy bodies, and this other condition called primary autonomic failure, which is also thought to be a synucleinopathy. And they found, again, this to be pretty sensitive, meaning that the majority of the people who had the clinical diagnosis did have a positive test, and it was accurate in anywhere from 95 to 97% of the time. So that's pretty good. That's very good, actually. Again, very good results from this particular test. Something to point out is that as of right now, this test is not able to differentiate multiple system atrophy from Parkinson's disease or dementia with Lewy bodies. It's positive on all of them, which is a little bit different than the other test. In addition, it can only look for synuclein. We still haven't figured out a way with a skin biopsy to identify people with amyloid, such as folks with Alzheimer's disease or tau. And so this one can only be done to identify synuclein, but it's pretty darn good. All right. So with these different approaches, colleagues and mine have started to think about whether or not it's possible to conceptualize whether we can start having a definition of these neuronal synuclein diseases based on people being biomarker positive. And again, this is just an initial framework, a conceptualization of asking the question, with these really good biomarkers coming out, can we start to put together a framework where we can, in parallel with the clinical diagnosis, also be able to say, well, what if somebody doesn't perfectly fit that clinical diagnosis of dementia with Lewy bodies? What if they don't have visual hallucinations yet, but they just have cognitive impairment and a positive synuclein test? Are we ready to call that person that they have a disease or not? And this is what we've just started to put this together. Again, it's a framework, it's a conceptualization that's starting to think about how we might use these assays in the context first of research, so that we can understand these things better, so that potentially you could imagine in a clinical trial using one of these biomarkers to include or exclude people to make sure everyone in the study has the biology that we are trying to ultimately study. Okay, so I'm going to throw out a couple of, I sort of did my own personal QA with a couple of questions, and I'm going to answer my own questions for a few minutes, and then I'm going to hand it over to Keith to ask me the questions that I did not get to. So there's a couple of ones that I wanted to make sure I really throw out there. So number one, should I get this test done? So if you have a diagnosis of Lewy body disease, should you get this test done? Not necessarily. Okay, so why not? So for people who already have a diagnosis of Lewy body disease, and the physician is really confident about the diagnosis, the information of the test will not necessarily change anything about your current treatment of care. And both tests are still a bit invasive. So a skin biopsy, again, is pretty simple, but it's not nothing. And then a lumbar puncture, again, pretty simple. We do it in clinic all the time, but it's not nothing. But costs are sometimes difficult. It's not always covered by insurance. And so I don't necessarily recommend it. And I would say in my personal clinical practice, I've done this I have a research cohort here at Stanford of over 500 people, and we've gotten who had donated their cerebral spinal fluid for research, and we've tested all of them. Whereas I've only done about a dozen of these tests in the clinical setting. So I only do this when I really am unsure about the diagnosis, and most importantly, when the test results will make a change or a difference in what I would do for the patient as far as their medications. So I don't think people should run out and get this test to confirm their diagnosis if their doctors are pretty confident about their diagnosis. We are not saying that people should go and get this test right now. They're still invasive. They're still not necessarily covered by insurance. So just think about that. Okay. We also need to understand more about that frequency of false negatives and false positives. So as I showed you, there was that one person who had very clear positivity at autopsy whose CSF test was negative. And we know some of those skin biopsy ones were also negative. I know the folks doing the research are now doing some comparisons with autopsy studies. So it's not 100%. And so we need to understand more about that frequency so that we know how to interpret. If I had someone who I was absolutely convinced clinically had the diagnosis of dementia with Lewy bodies, and they got the test and it was a negative, I would wonder, is this a false negative? And so we need to understand that more. And the only way we can do that is with research data. Is it 1 in 100? Is it 1 in 1,000? Is it 1 in a million? It's hard to know at this point in time. Okay, so if you shouldn't get the test, why am I making such a big deal about it? This is a really big thing for research. So it's a really exciting finding that has energized a lot of pharma companies and startups to be interested in drug development for people with Lewy body diseases and Lewy body dementia in particular. And that is awesome, because that means that this will bring more and more therapies and treatments to patients, and that's what we ultimately need. I do all of this biomarker research for one reason and one reason alone, and that is because I believe that having more accurate diagnoses for individuals in clinical trials will help us to get better therapies to patients tomorrow. Everything I do in my research lab is so that we have something better to offer patients in my clinic, and this has helped. I have had more conversations with pharmaceutical companies and startups in the last year about doing research in Lewy body diseases than I have in the last 10 years. And it's really important because companies will be more confident that the people they enroll in their clinical trials actually have the disease that's being targeted by their drug. And this lowers their risk and makes them more likely to invest in these therapies. So this is a really big deal for us getting better therapies to patients. So if you have the test and the result is negative, what does that mean? Whoops, I went too far. Shoot. Okay, we're gonna go this, then we're gonna go back to the other one. Okay, number one, it means that the test was, it could be a wrong test. It could be a false negative. It could be the same thing as my first COVID test, right? And so you have to keep that in mind. Or it could mean that you have a neurodegenerative disease, but just not the kind that's caused by a Lewy body. And right now, we need more research to understand the frequency that these two things happen. And so we need people who are willing to engage in research, have the biomarker test done, and commit to doing a final autopsy at the end. Because even as we get better and better and better, we still need to understand how these really do or do not predict final autopsy. Okay, I'm going to go back to this one. Sorry. Okay, so how is this biomarker different from a DAT scan? So some of you might have heard or even had a DAT scan. A DAT scan is a scan, a brain scan that looks at the dopamine production. So those neurons that I showed you in the brain that have the Lewy bodies in them, that have the neuromelanin, those are the nerve cells in the brain that produce the neurochemical dopamine. And so when those nerve cells die, you have less dopamine production in the brain. So that is actually a really good biomarker as well for the loss of dopamine production and the death of those dopamine producing neurons in the brain, which happens in people with Lewy body dementia and with people with Parkinson's disease. However, and so you get in someone with Lewy body disease, you have alpha-synuclein causing Lewy bodies in these nerve cells, and you get this. This is what a dopamine scan or a DAT scan looks like, where you have this reduction in the amount of dopamine production. This whole area should be red here, where it's just kind of that tiny little red dot up there. However, there are other proteinopathies that cause these DAT scans to be abnormal. So multiple system atrophy also causes an abnormal DAT scan, but it can do so by having this alpha-synuclein in different types of cells, not Lewy bodies, but rather having this alpha-synuclein deposits in these glial cells. And progressive supranuclear palsy and cortical basal degeneration also cause an abnormal DAT scan, but they do it by the neurons dying associated with tau deposits. So the problem with a DAT scan is it can just say that there's a reduction in the dopamine production, presumably because of the death of the dopamine producing neurons, but it can't say which protein was associated with it. So that's always been the limitation of a DAT scan, is it can't tell the difference between all of those different neurodegenerative diseases caused by different proteins on that first slide I showed you. So that's the issue with DAT scans. So this is true. This is good, but you can see this in multiple neurodegenerative diseases, whereas the synuclein specifically, you can't tell whether it's Lewy bodies, these glial inclusions, or the tau based on the DAT scan. Okay. So finally, I think these are my last two questions before I'm going to pass it over to Keith. Should people at risk for Lewy body disease or family members get the test done? No, or only do so in a research setting. As I said, right now, this test has not been well validated in people who don't have any symptoms. We are working on this very, very aggressively in lots of these aging research studies. But right now, if someone has absolutely no symptoms whatsoever and their test is positive, we don't know if that is a false positive or a true positive. We need more data to support that. And most importantly, we don't know the relationship between a positive test, assuming it's a true positive, and when they might develop symptoms. So let's say someone's 60 years old and they don't have any symptoms and they happen to get this test done and the synuclein's positive. I don't know whether they're going to develop symptoms in 1 year, 5 years, 10 years, 20 years, 40 years. We don't understand that relationship. time is a really important part of this. And this is a really critical question that we need to answer. And the only way we're going to answer this is through research, which is why my last slide here will be a plug for consider participating in research studies focused on understanding biomarkers that can help us find treatments for Lewy body dementia. And it really is true that actually 30% of all clinical trials fail to recruit a single person. 85% of studies face delays due to limited participation, and fewer than 10% of patients with Lewy body dementia ever take part in a trial despite overwhelming interest in working with scientists to help speed treatment breakthroughs. So if the study that Keith talked to you about, and I'm actually not part of it at all, so I'm not even plugging my own study here, if the one that Keith talked to mentioned at the beginning isn't the right thing for you, there are many, many other options for good research studies to be in. And in fact, if you don't have dementia with Lewy bodies and your loved one does, and you are a healthy person who doesn't have any neurological disease, we need those people to enroll in clinical trials as well. Because as I said, we need to understand the frequency of false positives in people who don't have any neurological findings in order to truly understand the validity of these tests across a wider enterprise. And so this is a really important area. So thank you all for your time. This is my final slide, my wonderful research lab, my wonderful supporters, and I will end there and pass it back over to Keith. I'm going to unshare my screen so that you can see us talking. So let me just do that. Stop share. Okay.
Yeah, thank you very much, Kathleen. So that was, and you actually, I really love what you did at the end there with answering some of the questions, sort of questions you had sort of pre-thought of. Let me just deal with these right up front, because you did get a lot of questions that fit into some of those areas. Before I start going through these questions, though, I would like to echo your plug for participating in clinical research. There really are a lot of terrific studies out there, you guys, that you can participate in. I would encourage you, we did drop the link for the CIND study into the chat. Take a look at that, because it's focused specifically on the topic here today. But there are also medication trials. There are trials of caregiver interventions. I mean, you name it, researchers are interested in it, trying to optimize quality of life for people with DLB and their care partners. I think one of the easiest ways to find those studies is just go to lbda.org, and across the top of our website, you'll see a navigation bar. Just click on research, and you'll be able to see a page there that has all of the research studies that we're aware of that you might be able to participate in, listed out there for you, along with the study teams and the locations that may be near where you live. There are also lots of studies that don't require clinic visits. So even if you live somewhere where you feel like you're not close to a major medical center, there probably are still also studies that you can participate in. So we do urge you to go take a look. Without any further ado, though, I'm going to jump into some of these questions. We've got a lot of really great questions for you. So some of them are specifically on the topic of these biomarkers, and some are a little bit more general, but might still be helpful for people understanding the content today. So one of them is, I recently heard LBD described as Alzheimer's and Parkinson's joined to produce LBD. What is your position on that description?
Yeah, that's a great question. So I showed in that very first slide with all of the different neurodegenerative diseases, I showed them as being totally different and discrete things. And that might have been a little bit simplistic. It turns out when we actually look in the brains of individuals who have passed away and given their brain for research, that actually a good number of people have more than one of those proteins in their brain. And it used to be thought of that was the exception, but as more and more of wonderful people have donated their brain for research, and we have, again, the order of, hundreds to thousands of samples, we really understand that actually probably 50 to 70% of individuals have more than one thing in their brain at the time that they died. And actually, people with a clinical diagnosis of dementia with Lewy bodies are some of the higher percentage groups that have more than one thing going on. The most common second thing going on in their brain, in addition to the Lewy bodies, is some of that Alzheimer's type pathology with the amyloid and the tau. And it's one of the reasons why in fact, in our research cohort here, we sent about 100 samples of individuals with the diagnosis of Alzheimer's disease, dementia with Lewy bodies, and Parkinson's disease for amyloid, tau, and for that synuclein test. And we found about 15% of the individuals with Parkinson's disease or dementia with Lewy bodies had positive amyloid. But if you just looked at the dementia with Lewy body group, it was higher. It was more like around 40+%. Interestingly, we also found about 10% of individuals with the diagnosis of Alzheimer's disease had a positive Lewy body test. So in fact, it's the other way too. And so I think that it is, we're gonna, now that we can test people in life for these different proteins, I think we're gonna understand a lot more about how there are multiple things going on in the brain, and that will be very important for clinical trials and ultimately therapy. Because if someone has the synuclein and the amyloid, they might be a good candidate for two different types of therapies. Whereas if someone just has the synuclein, they might not be a candidate for one of the other types of therapies. So this is a really important thing for us to figure out.
Great, thank you. And actually, one person had exactly that question. Can you have both AD and DLB at the same time? And so I'm just going to mark that one as answered. Yeah. And now here are some questions that are more looking sort of, behind the curtains, or maybe asking you to make some predictions. And so, just with the understanding that no one has a true crystal ball, when do you think the new biomarkers, both skin and CSF, or one or the other, will be added to clinical diagnostic criteria?
Yeah, so the reason why we put out that first paper that I showed you, which was just published earlier this month, was our first attempt at putting the biomarkers in a framework for a diagnosis. But I think right now, again, we meant it only for research studies. And we believe that there's probably going to be this period of time where we'll have this parallelism where, when I'm in clinic, I make the diagnosis based on the clinical diagnostic criteria. When I have my research hat on, I'm categorizing people according to the biological markers. Those two things will continue to progress and merge more and more as we understand more. But I'll be honest, I think that for something to truly be in the day-to-day clinic for a diagnostic, it would really be ideal for it to be available in something like a blood draw. Because we all get blood draws, right? I mean, I'm of the age that I get my cholesterol checked and my thyroid checked whenever I go see my doctor every year. And that's something that's done very, very frequently and is easy to do. I think we're a couple of years away from having these tests in blood, but I actually very strongly believe that we will be able to get it in blood eventually. I know a lot of great researchers working on it. So I think we're going to go through a little bit of an odd period of duality where the majority of people will be diagnosed based on their clinical diagnosis, but we'll understand more and more about the biological diagnostic, the best way to biologically diagnose people moving forward.
Great, thank you. And so you already asked or answered rather about, should a person go get a test if their family member has DLB or PDD, et cetera. But we have questions here about insurance coverage as well. What can you tell us about whether Medicare or private insurance might cover these tests?
Yeah, so this is a really rapidly moving area. And so I actually can't with 100% confidence say that what I'm going to say is still true and whether it's going to be true tomorrow. So I know that these companies are very actively working on getting insurance coverage. And I know in some situations people have been able to, but I don't know for sure. And I think it's still a little bit up in the air whether they will be covered by insurance from a clinical perspective. They are so new, and sometimes it takes like a year or two for the process to go through to even get evaluated for insurance coverage. So I actually don't know. I lean on the side of no, but they might be in some situations yes at this point. And I know both companies are working very hard to make sure that that is a definitive yes in the future.
So rapidly changing.
Rapidly changing, yes.
Trying to move in that direction.
Yeah.
So this is an interesting question about the, I assume this is specific to the skin biopsy. And the question is the distance from the brain or the amount of synuclein detected in the skin, are either of those significant in determining the probability or accuracy of a DLB diagnosis?
It's a really great question. And I'd say as of right now, we do not know. So typically for the skin biopsy, they're requesting the clinicians to take 3 biopsies, one close to the neck, one on the trunk, and one on the leg. And it seems like the pattern, like if it's just, it seemed like the one on the neck might be the most accurate, but at the same time, I have one person who came back and the one on the leg was the only one that was positive. So it's not always the case. And so this is something that is really important for us to investigate further. There also, it might be that the pattern or the amount of fluorescence they see under the microscope has clinical meaningfulness. As of right now, that is all you know, good hypotheses that researchers are generating. I would not say it's something confident enough that it's clinically meaningful, but we are trying to figure that out.
Great, thank you, thank you. And then here's a question about the stability of the tests. If you get a positive test result one time, how likely is that to remain stable over time?
Yeah, so that's there's 2 questions there. There's test retest, meaning if I took the exact same sample and tested it and then took a little more of the sample and tested it, would it stay positive? And the test retest for both of these is pretty high. So if you do the same sample, we are just now starting to collect longitudinal samples. So that's if I test today and then I retest again in 6 months or 1 year from now, are you going to have, so does the biology of the person change in a way that the test becomes negative over time? A lot of you, again, going back to that COVID experience, might have, some people's COVID antigen test became negative sooner than others. And so how does time play into it? So we don't have a lot of longitudinal samples on patients. I actually just came off a call looking at longitudinal CSF samples in individuals with newly diagnosed Parkinson's disease, and the data looked very good, but we only had about 40 people where we truly had longitudinal samples from. So a lot more needs done to understand that piece of it. And honestly, longitudinal biological testing is a really hard to recruit for. So if anybody is willing to do longitudinal samples, bless you, because that kind of research is really necessary, and I know it's hard for people to do.
So I think we have time for one more question. And so I have to select from among all of these, but I'm gonna end with this one. With the skin biopsy, can it be taken from anywhere on the body, or does it have to come from certain areas?
Yeah, so the key to any type of assay is that it's done as consistently and regularly as possible. Again, think about your experience with the COVID test, right? They gave very clear instructions about exactly where the swab was to go and how many times you should rotate it and all that kind of stuff. And it probably was okay if you didn't do it perfectly, but the more you did it according to the exact instructions, actually, the better the accuracy was. And so, and it's just a matter, they have to prove that, doing it if there's variance, that it has the same accuracy. So I think it's very important right now that people do it exactly the way that the company is saying that it needs to be done, as close to the exact type of procedure as possible, because that's what their data has shown. And so until we know something differently, I would make the assumption that if you do it differently, it's not going to be as accurate.
That makes sense. Great. Thank you very much. So thank you all for attending today and for your questions. Unfortunately, we are out of time. We will make an attempt for staff to answer as many of these questions that we received as we can, if your question was not answered today. I'd also like to thank Dr. Poston for her time and for sharing her presentation with us today, and also for answering your questions. I'd like to again mention the SIND study, since that's on the topic of today's webinar, if you are interested in research. But you can also look at other research studies, as we mentioned, and the links for that are in the chat. With that, we're going to give you a reminder here that you're going to receive an email in about 1 week. That email is going to have the link to the recording from today. We also invite you to check our events calendar frequently. You can find that at the bottom of our homepage on our website. And that has details for registration for upcoming events. If you're interested, you can go ahead and mark your calendar now for Friday, March 8th at 2 p.m. Eastern Time. And at that time, we'll be joined by Dr. Jory Fleischer of the LBDA Research Center of Excellence at Rush University. And she will present Unpacking Parkinsonism, Understanding Movement Changes in LBD. One final reminder right before we go here is please do take a few moments at the end of the webinar to complete the survey that will launch after you drop off. And again, that just helps inform our content for the future. So we hope to see you all again the next time, and thank you.
Thank you.
Yep.